For nearly half a century, advances in medicine and biotechnology have been hampered by one very tough and solvable problem. How do proteins fold up? But now, thanks to amazing advances using AI, this grand challenge is considered solved, opening the path to an entirely new era of AI-assisted computational biology. And so therefore, in this video, I want to cover one of the most anticipated machine learning models of the past years, which has finally been released in all its glory. I'm talking about DeepMinds' latest version of the AlphaFold protein folding model. So for those of you who are not very familiar, let's listen to David Baker, one of the gurus in the protein world, explaining what proteins really are.
Proteins, some of which you see inside a cell here, carry out essentially all the important functions in our bodies. Proteins digest your food, contract your muscles, fire your neurons, and power your immune system. Everything that happens in biology, almost, happens because of proteins.
That's right. Proteins are everywhere. Now, if you don't yet have a good intuition about how these fascinating molecular machines work and how they're created from the genetic material in your DNA, I really recommend you watch my previous video on molecular biology first, because we're going to be building heavily on those concepts over here, and this video will just make a lot more sense if you have the necessary mental framework to place all of these ideas. Alright, so in this video, I'll start by quickly introducing the historical events that led to the 5-decades-long quest to solve the protein folding problem. We'll then briefly revisit the original AlphaFold 1 architecture before diving into what's arguably one of the most impactful machine learning models of this decade, AlphaFold 2. We'll explore its full network architecture and training principles, and after that we'll zoom out a little bit and look at what this breakthrough means for the fields of computational biology and protein design. So, are you ready to dive in deep? My name is Xander, and welcome to Archive Insights. So what is the protein folding problem? Well, in his acceptance speech for the 1972 Nobel Prize in Chemistry, Christian Anfinsen famously postulated that, in theory, a protein's amino acid sequence should fully determine its structure. This hypothesis then sparked a 5-decade quest to computationally predict a protein's 3D structure based on its amino acid sequence. A major challenge, however, is that the number of ways that a protein could theoretically fold is just astronomical. I mean, in 1969, Cyrus Leventhal noted that it would take longer than the age of the universe to enumerate all possible configurations of a typical protein by brute force calculation. Yet in nature, proteins fold spontaneously, some within milliseconds, a dichotomy often referred to as Leventhal's paradox. This paradox then led to the assumption that instead of trying out different combinations at random, well, nature must be using predictable folding pathways to arrive at the native state of a protein so reliably and rapidly. And ever since then, scientists from all over the world have been trying to model these folding pathways using advanced computer simulations that try to approximate the physical interactions underpinning the folding process. Now, the hard truth, however, is that after decades of incredible work on physics-based modeling tools like Rosetta, for example, in many cases, the accuracy with which they can predict the 3D structure of a folded protein is simply not good enough to be practically useful. Enter deep learning. Now, over the past few years in AI, we've witnessed a surge of learnable programs that excel at challenging problems that are too complicated for us humans to write down an explicit solution for. Now, the main idea here is that these systems learn a differentiable program purely from example data. A good analogy here is what happened a few years ago in chess and Go, where traditional engines like Stockfish that were manually engineered by decades of handcrafted rules were surpassed by end-to-end trainable deep learning architectures like AlphaGo and AlphaZero. Now, the training data for AlphaFold comes mainly from what's called the Worldwide Protein Data Bank that stores a freely accessible archive of all known protein structures, each of which is the result of a very expensive measurement using things like X-ray crystallography and almost a PhD worth of work to analyze all the data and determine the final structure. So in other words, determining the structure of a protein has traditionally been very challenging and very expensive, which is exactly the problem that AlphaFold addresses. Now, before diving into the latest DeepMind paper, I quickly want to highlight the original AlphaFold 1 architecture, which won the CASP13 competition at the close of 2018, because a lot of the ideas from that earlier work have been refurbished in the new architecture. After that, we'll look at the updated version of AlphaFold that won the CASP14 competition in 2020 with incredible results. So AlphaFold 1 is actually a fairly straightforward architecture. The entire pipeline starts with an intricate feature extraction process whereby external databases are used to find similar protein sequences to the query sequence to form what's called a multiple sequence alignment. So, all right, what is a multiple sequence alignment? Well, from studying different organisms over the course of evolution, we know that the three-dimensional structure of a protein is much more stable over time than the genetic sequence that encodes that particular protein. So, in other words, mutations to the specific amino acids that form a protein are quite common in nature, but changes in structure are much more rare. And if you think about it, this actually makes a lot of sense. The reason is that most living organisms descended from common ancestors. So the proteins performing all these biological functions in their bodies, well, they will have very similar shapes because they need to keep carrying out the same functions like, transporting oxygen or breaking down sugar molecules. So if a mutation were to occur in the DNA that drastically affects the shape of a folded protein, well, then that protein would most likely stop working properly, and there's a good chance that the organism would simply die off by natural selection. And so the only mutations that survive are the ones that don't really affect a protein's structure. If you then compare the sequences of a particular protein over multiple species that descended from a common ancestor, well, you end up with various sequences that should have strong structural similarities. And this is exactly why multiple sequence alignments carry such relevant information for the protein structure prediction task. And yes, I know that it's tempting to try and solve the protein folding problem using just the single sequence information of the target. This is what happens in nature. But the reality is that we just have a ton of sequence data sitting in these databases, which provides valuable information about protein structure. So why not use it? All right, so the first version of AlphaFold took the target amino acid sequence and it then starts crawling all these databases to find evolutionary matches in related species of the animal kingdom. This MSA information is then combined with some additional input features, processed and reshaped into a two-dimensional array and fed into a deep residual convolutional network that's very similar to what we use for things like image classification. And the main target output of this ResNet is the distogram of the actual protein that we're trying to predict. All right, so what is a distogram? Well, a distogram is very simply a 2D array of distances between every pair of residues in the protein. So at row I and column J, you will find the distance between amino acid I and amino acid J in the 3D shape of the folded protein. So essentially, a distogram is a two-dimensional projection of the 3D shape of a protein. And since the distance from I to J is the same as from J to I, the real distogram, well, that will always be a symmetrical matrix around the main diagonal. Now, why would you use a distogram instead of directly predicting the 3D coordinates, you might ask? Why take this intermediate step? Well, there's actually a lot of reasons someone might want to do this, but one simple and important one is that the distogram is invariant to rotations and translations of the protein. So if you take the 3D structure and you move it around and rotate it, well, the relative distances between the amino acids stay the same, and so the distogram is the same. So this makes your target much easier to learn, since you don't have to figure out some canonical way of aligning the protein every time you do a structure prediction. And now, rather than trying to directly predict the target distogram, AlphaFold actually predicts a distribution over distances. So for every pixel in this 2D matrix, AlphaFold will predict a distribution over 64 possible distance bins, which is incredibly useful because it provides an estimate of how certain the model is about the distance between two particular residues. And then finally, to get from this predicted distogram distribution to the final 3D shape, AlphaFold 1 used a gradient descent approach. Essentially, a physical model of the protein chain is constructed, which is parameterized by the phi and psi angles between every two consecutive residues. And this backbone geometry is then iteratively refined using a scoring function that combines the predictions of the neural net with a set of physical simulated forces to figure out the most likely protein conformation according to the model. Now, I know that I skipped over a lot of details here, but that's the essence of AlphaFold 1. You first collect a multiple sequence alignment, you then predict the distogram using a ResNet, and then you optimize the protein backbone using the predicted distogram in combination with a set of physical forces. So now let's transition to the updated version, AlphaFold 2. And now, while DeepMinds AlphaFold has definitely been at the forefront of protein structure prediction for the past few years, I can't make this video without at least mentioning some of the other amazing effort in this space. Some examples, and I can't mention everyone, include the amazing open source work by the Baker Lab at the University of Washington in Seattle, and more recently, some incredible efforts to replicate the advances of AlphaFold by the open source community. And while DeepMind refrained from publishing their paper for almost a year after first announcing AlphaFold 2, it's very likely that they were feeling the building pressure of these ongoing efforts breathing down their necks and almost forcing their hand into publishing their amazing work before getting scooped by the broader community, because a lot of these ideas were also being explored by other groups. So this is another great example of how a lively open source community is a really good safeguard against building monopolies. Alright, so now that we've set the stage, it's time to dive into the machine learning architecture of AlphaFold 2. Hold on to your hats because it's about to get a little technical. So AlphaFold 2 consists of three essential building blocks. The first part of the architecture is the pre-processing stage, where the input sequence is used to query additional information from external databases. This information is then mapped into an MSA and a pair representation, which are refined by what's called the Evoformer, a 48-layer deep transformer-like network that uses attention mechanisms to update the MSA and pair representations. The outputs of the Evoformer are then processed by the third component of AlphaFold called the structure module, which basically transforms the abstract representations formed by the Evoformer into concrete three-dimensional coordinates of the protein geometry. And with that overview in mind, let's take a look at each of these components separately. So given an input chain of amino acids, AlphaFold first creates a multiple sequence alignment, just like in the first version, by searching for similar evolutionary sequences in a genetic database. Additionally, though, AlphaFold 2 will also search for structural templates, which are known protein structures that have similar sequence parts as the query sequence. Now, during training, these structural templates are sampled in such a way that the model sometimes receives very bad templates or no templates at all, which has the effect that the network cannot just rely on copying a good input template to get an output structure, but it really is forced to learn from the sequence information instead. AlphaFold then integrates all this information from the input sequence, the multiple sequence alignment, and the template structures into a pair representation and an MSA representation, which will be used for further refinement. In fact, because there is so much external data available, this pre-processing pipeline is usually repeated a few times, leading to different versions of the pair and MSA representations, which can then be averaged together before flowing into the structure module. So once the MSA and pair representations are formed, they are forwarded to the Evoformer. The Evoformer is a 48-layer deep network that contains two separate flows of information. One for the MSA representation on top and another flow of information for the pair representation at the bottom. All these 48 blocks have individual parameters, so this is not a recurrent network unlike the structure module that we'll discuss in a bit. Each block has an MSA and a pair representation as input as well as its output, and it processes them with several layers. Now, the layers inside the Evoformer, well, they actually have a lot of very clever technical tricks that make it particularly suitable for updating the protein representations coming from the input stage. And the main idea here is to allow the network to iteratively refine the information from the MSA and pair representations by allowing the two modalities to exchange information, which essentially enables direct reasoning about the spatial and the evolutionary relationships in the protein. So to translate this in plain English, the idea is that the multiple sequence alignment contains information about the evolutionary trajectory of mutations for the particular protein in question. But in order to extract all the relevant information from this MSA, well, you would have to know something about the structure that you're dealing with. And this is what the pair representation tries to do. So in essence, these two flows of information are just interacting together to solve an evolutionary puzzle about the input protein. Now, I'm not going to go into all the technical details of how exactly the Evoformer does this, since there's just way too much stuff, and you really need to read the supplementary to the paper if you want to dive deep in the rabbit hole. But very briefly, the MSA stack uses an attention schema called axial attention, whereby alternating layers apply row and column attention sequentially. The idea being that the row-wise attention allows the model to reason about information from each of the aligned sequence separately, while the column attention lets the model reason about relationships between the aligned residues in different sequences of the MSA stack. Additionally, the row attention is influenced by a branch coming from the bottom, allowing the information from the pair representations to flow into the MSA stack. Likewise, another branch of the network goes back down to the bottom flow to enable an update of the pair representations. Now, interestingly, this operation is applied within every one of the 48 blocks in the Evoformer, enabling continuous communication between the evolving MSA and pair representations. Now, the pair representations themselves are updated using another very specific mechanism that essentially tries to make the 2D representations consistent with the three-dimensional structure that they represent. And to understand what's happening here, consider that the purpose of these 2D representations is to encode things like inter-residue distances. But there's a bit of a problem here. Remember that the feature vector at position ij should encode the distance between residues I and j. So now imagine creating a triangle in 3D space going from I to j to k. Now, because these two-dimensional features are the output of the neural net, well, it's very likely that the predicted distances in the 2D feature map are actually incompatible with any possible physical triangle in 3D space. And this is why this part of the network allows the 2D feature map to update itself in such a way that it becomes more consistent with an actual three-dimensional structure. Again, if you want to learn how exactly AlphaFold does this, you should definitely check out the paper because there are some very clever tricks here. But so that's the general idea behind the Evoformer, to gradually mix information between the MSA and the pair representations to prepare for structure generation in the structure module. So the final piece of AlphaFold is the structure module. Now, this module transforms the abstract representations that come from the Evoformer stack into a concrete three-dimensional protein geometry. It does this by passing the first row of the final MSA representation from the Evoformer through 8 network layers that all share the same weights. So this is essentially a recurrent network that iteratively refines the spatial geometry of the protein. Evoformer's pair representation is also used in this process. Now, the structure module operates on a concrete three-dimensional backbone, which is represented as a set of independent rotations and translations for each residue with respect to a global frame of reference. So this is actually pretty fascinating because it completely ignores the fundamental constraints that we know to be present in nature, and yet it appears to work really, really well. Now, in the first iteration of the structure module, the 3D coordinates are not yet known, and so everything is initialized to an identity transform, which the authors fittingly call the black hole initialization. It simply means that at the start of the structure module, all the residues in the protein are located at the exact same point, which is the origin of the global frame, and they also have the exact same orientation. Now, after this initial black hole initialization, the residue gas is then updated iteratively in 2 stages. First, a novel geometry-aware attention operation called invariant point attention, or IPA, is used to update the sequence representation without changing the 3D positions. Then, an equivariant update is performed on the residue gas using this updated sequence representation. Now again, the invariant point attention mechanism involves a whole bunch of very clever tricks that were specifically designed for the purpose of protein structure refinement. In a nutshell, the invariant point attention augments information coming from the local frame of each residue such that the activations are invariant to rotations and translations in the global frame. So this idea is very related to all the recent efforts around spatial equivariant transformers and graph neural networks. If you want to learn more about this, I'd highly recommend this blog post from Fabian about equivariance in machine learning. It's an amazing read. Another interesting note is that the proposed frame rotations are represented using something called quaternions rather than the usual 3x3 rotation matrices that we're all used to. Now, I suspect there's a bunch of different reasons for this, but quaternions actually do provide some interesting benefits over regular rotation matrices. For example, they can be represented using just 4 numbers instead of the 9 numbers that you need for a 3x3 rotation matrix. Additionally, interpolation between various angles is a perfectly smooth process where you don't have any special edge cases like you have for angles where 0 degrees and 360 degrees is essentially the same angle, but it's a different numerical value. So this could make quaternion parametrization much easier to optimize with gradient descent. And finally, AlphaFold also fixes the first of the 4 quaternion numbers to be 1. And what this does is that it biases the learned rotations towards very small values, which is really desirable if you have an iterative updating scheme. If you want to learn more about quaternions, 3 Blue 1 Brown has some amazing videos on the topic. They're highly recommended. So without going into too much detail here, the main idea is that by iteratively updating the sequence representation and the backbone transformations, the black hole initialization of the protein is gradually adjusted until the final backbone geometry emerges. And then finally, a shallow ResNet is added on top of the final abstract representations for each residue to predict the torsion angles for all the atoms in the sidechain, which ultimately leads to the 3D coordinates for every atom in the protein structure. And voila, that's it. That's the full network architecture of AlphaFold 2. Now, let's take a look at how this beast was actually trained. So AlphaFold was trained using a combination of 4 different loss components, the most important of which is what's called the frame-aligned point error, or FAPE loss. Now, what FAPE does is it essentially compares the atom coordinates for the predicted structure with the ground truth structure, but it does this many times over using a different alignment for every local residue frame. And this has the effect of putting extra importance on getting locally consistent structure for every residue rather than only focusing on the global coordinate accuracy. The idea here is that it's very, very hard to get all the atom coordinates perfectly right, and more often than not, you want the model to focus on correct substructures first, and only then start worrying about global placements. Now, aside from this FAPE loss, there are a number of auxiliary losses. First, the final pair representation is linearly projected to a distance distribution scored with a cross-entropy loss. So here, the Evoformer stack is being supervised by the target distogram. Second, random masking is applied to the input MSAs, and the network is tasked to reconstruct those masked regions using the output MSA representations with a loss very similar to a model like BERT. So here, the tricks from unsupervised language pre-training have been applied to the input MSA stack, which essentially forces AlphaFold to learn about the evolutionary relationships between the sequences in the MSA. Third, the single representations at the output of the structure module are used to predict per residue LDDT scores. What this means is that AlphaFold tries to predict how well it thinks it's modeling the position of every residue. And this is a very useful output because it shows you where AlphaFold is very certain about its predictions and where it isn't. And finally, there are two minor auxiliary losses that are used to better predict the sidechain positions and avoid structure violations whereby two atoms would be at the same place, for example. And now, the main purpose of all these different losses is to attach an individual objective to each major component of the model, which serves as a guide during training for the purpose of each subunit. Now, because the backbone is modeled as a residue gas, there's always a possibility the network could predict atom coordinates that clash, meaning that two residues are predicted closer together than is physically possible. So in order to resolve any remaining structural violations and clashes, the final predictions are relaxed with a restrained energy minimization using the amber force field. Now, towards the end of the paper, there's a very interesting graph showing how different elements of the AlphaFold algorithm affect the final performance of the model. And on this ablation study, you can see a few final tricks that I haven't mentioned yet. For example, AlphaFold used something called self-distillation to increase the size of its training data. Essentially, AlphaFold was first trained on all available protein structures in PDB, but then it predicted the structures of roughly 300,000 additional proteins with unknown structure. These predicted structures were then added to the training data, but only the residues that AlphaFold thought it predicted with high precision were used to perform gradient updates. Another trick used is called recycling, whereby outputs of the model are recursively fed back into the same modules, essentially increasing the depth of the model and giving it more iterations to refine the predicted protein structure. So, as you can see, AlphaFold is not the result of a single breakthrough idea that magically solved the protein folding problem. Instead, it's a collection of cleverly designed mechanisms and novel ideas that have been improved over many design cycles, resulting in this amazingly well-engineered system. And there you go, that's AlphaFold 2 in all its glory. So to end this video, let's briefly zoom out a little bit and talk about the bigger picture. I mean, is protein folding really solved now? Well, yes and no. First of all, let me state this very clearly. AlphaFold is a tremendous engineering achievement by some of the most talented AI researchers in the world. The model is able to predict the structures of most single-domain proteins in biology with incredible accuracy, and it's undeniable that this will herald a completely new era in computational biology, where AI will be used as an indispensable tool for designing new drugs, understanding disease pathways, fighting viruses, and much more. At the same time, however, AlphaFold's accuracy isn't always perfect. For example, many processes in biology rely on large protein complexes, whereby many different proteins come together to form complex molecular machines, which are currently outside of AlphaFold's capabilities. There's also the notion of generalization. I mean, AlphaFold was trained on a dataset of proteins that naturally occur in biology, but we don't really know how good its predictions will generalize to completely new de novo proteins that we might want to design with these new tools. Additionally, most proteins function by changing shape with respect to targets they bind to. And these molecular dynamics, well, they're not captured by AlphaFold's static view of protein structure. Being able to model the flexibility of a real protein in these different conformations will be crucial to study protein-protein interactions. That being said, though, it's very likely that the ideas proposed by the AlphaFold team can be extended to those new challenges given the right types of training data and engineering efforts. All in all, I think many people will agree with me when I say that this is probably one of the most impactful machine learning models of this decade. So the fact that DeepMind has finally published their paper and released their code, including pre-trained model weights, is an incredible gesture towards the scientific community. In fact, we're now living in a world where you can simply run AlphaFold, the world's most advanced protein folding model, directly in your browser using Google Colab with zero setup, giving you state-of-the-art structure predictions for any protein you're interested in. I mean, honestly, that's pretty mind-blowing. And with that, I would like to thank you very much for watching till the end of this video. Please consider supporting this channel on Patreon. Making these videos is a lot of work. Check out my Penside page if you need one-on-one meetings to discuss your own projects. But most of all, say thanks to the billions of active proteins keeping you alive right now. Have an amazing day.